science geeks. Welcome to Late Night News, and I am your favorite host, Areeb Siddiqui. Today on Late Night News, we have an interview with the revolutionary man, the man that can change the world, the one and only Dr. Masahito Tachibana. He is the man that has revolutionized the world of cloning through his discoveries in the field of embryonic stem cells. You better believe it, that someday in the distant future, there will be a clone of you sitting in front of you. Anyways, back to the interview. Mr. Tachibana, it is a great honor to have you with us.
The pleasure is all mine.
So, Mr. Tachibana, I have heard that you have made a massive accomplishment in the world of embryonic stem cells. Would you mind giving us some insight into your research?
First, let me start with basics. Embryonic stem cell research has been going on for years, and I guess we were the first ones to find a real breakthrough. Pluripotent stem cells possess the ability to divide for long periods and retain their ability to make all cell types within the organism. The best known type is an embryonic stem cell. So basically all your skin cells, nerve cells, muscle cells were all first embryonic stem cells, but then due to differentiation, the stem cells turned into specialized cells. Now, our mission was to somehow get to stem cells by producing embryos, since stem cells are found in the development stages of an embryo. Now, the thing with stem cells is that stem cells from a different body may not work in another body. So we need stem cells that are genetically identical to the body. In order to do this, we have a process called somatic cell nucleotransfer, in which the nucleus of a patient's somatic cell is transferred to an empty egg, meaning an egg without a nucleus. Then the embryo starts developing with the genetic information of the patient and starts producing stem cells that belong to the patient. Although this technique was working perfectly with animal eggs, it was not working with human eggs. After installing the nucleus in the embryo started dividing, but it did not go past the 6 to 12 cell stage. And this is where we came in. We realized that the reason somatic cell nucleotransfer was not working with human eggs was that the oocytes were premature and not fully developed. The oocytes are premature eggs. So basically, in the past, we were experimenting with premature eggs, which resulted in a failure. But when we realized this problem, we started to use mature eggs, and due to this, we developed the embryo to the blastocyst stage, which is around 150 stem cells, enough to produce stem cells. So we basically made a fertilized egg grow to the point that there were stem cells.
Are these embryos viable to be real humans?
Well, as I was explaining earlier, the embryos created by the somatic cell nuclear transfer are supposed to turn into real human beings. This is pretty much like cloning. We're using the DNA of a person into making the embryo that is completely identical to that human being. So if the embryo were to grow, it would eventually turn into a clone of that human being. But till today, our experiments have only developed the embryo to the blastocyst stage.
Would this have any relation to the creation of Dolly the sheep?
Actually, this was the exact process that was used to clone Dolly, the sheep. Here's a little demonstration of what happens.
The transfer we'd seen before, but now we're going to see it as an example where a skin cell nucleus is going to be used to make an embryonic stem cell. So the egg cell has the nucleus removed. The nucleus from a skin cell is then injected into that egg. That egg, you'll remember, can then initiate development by dividing, making a blastocyst. Here's this important reprogramming phenomena going on, the details of which are still unknown. The cells now begin to divide, continue to divide to make a blastocyst with an inner cell mass, and then it's from the inner cell mass in that blastocyst that an embryonic stem cell can be derived.
So basically what happened was that the nucleus of Dolly was injected into an egg, and the egg developed to form Dolly the sheep. This is somatic cell nucleotransfer.
How is this vital for the future?
Well, if we can find a source of stem cells, the world of medical science could take a major leap. Imagine if someone needed some brain cells. Brain cells cannot reproduce. So if you have 100 brain cells, you will never exceed 100 brain cells. But brain cells sometimes die out, and there's no way to reproduce them. But through the use of stem cells, we could potentially turn them into a nerve cell or any other type of cell that could help in healing or rehabilitation. If someone was to suffer from heart disease, we could replace the dead or decaying heart cells to new heart cells formed from embryonic stem cells.
What are some previous benefits we have seen from stem cells? I've heard that it has helped people's sight.
This is why stem cells are very, very vital for the medical field. Stem cells have been used previously to treat eyesight loss. Stargardt's macular dystrophy is an incurable disease developed in childhood which causes gradual loss of sight. UK medics have conducted a surgery on 12 patients by injecting retinal cells that were formed from stem cells into the patient's eyes. If such use of stem cells can be used to treat a variety of diseases, it can be a revolution in the medical world, and this is what we're looking for.
So explain the controversy going on with your research with embryos.
Well, critics believe that it is unethical to use human embryos to experiment this research. To be honest, the embryos we receive are contributed by willing patients. If the patients are not willing to give, we would never ask for embryos. And the patients that provided the embryos should be honored that the embryos they gave are contributing to the medical field by possibly saving many lives today and many more in the future. In many countries, reproductive cloning is forbidden, but what we are doing here is therapeutic cloning.
What alternatives do you have for embryos that will please critics?
Many critics think that since cloning animals is dangerous, then cloning humans is also dangerous. So they've come with a new way to create stem cells. The technique uses skin cells and converts them to induced pluripotent stem cells through the use of proteins. Induced meaning persuasive or believable stem cells. Although this method is relatively cheap compared to embryonic stem cells, there are still a lot of questions about the quality of the stem cells produced.
Are there safety concerns with stem cells?
I'm afraid some research shows that the use of stem cells can be hazardous to patients. It is believed by some that stem cells could pass viruses and other diseases to the patient when they get a transplant. Some also believe that stem cells can turn into cancer cells. Research has shown in animals that the use of stem cells can cause malformed body parts. This could be one of the down effects of cloning because it can be dangerous. But at the moment, we have just started to explore the world of embryonic stem cells, and who knows, maybe we can find a solution to these problems.
Mr. Tachibana, do you believe in cloning people?
Well, frankly, yes I do. Through the research we have conducted, I believe that in the distant future, cloning would be very possible. At the moment, we have managed to create embryos. A bit more research and development could turn those embryos into full-fledged humans.
This is it for tonight, folks. Thank you, Mr. Tachibana, and see you next time on Late Night News.
Thank you for having me.